Good evening and welcome. Welcome to our exciting series of conversations with some of the most inspiring women at Imperial College London. I want to thank Pinky Lilani and Marty Wickstrom for their support and helping create this series and connecting us to their wonderful networks of friends. Welcome to all of you. Imperial College London is one of the world's greatest universities focusing on science, engineering, medicine, and business. We have some 20,000 students and 8,000 staff pursuing the forefronts of their fields. I'm excited to say that our research discoveries are ever more present in the news and in our teaching. Every day we learn new things from the youngest members of our community. Thus, in this series, we decided to connect some of our stellar women luminaries and their breathtaking career moments with the ideas and aspirations of our younger colleagues and students. And tonight we begin with transformative science and innovative ideas from across disciplines. So let me begin by introducing our luminaries. Molly Stevens is one of our most acclaimed academics. She is professor of biomedical materials and regenerative medicine in the Department of Materials and the Department of Bioengineering. She's the research director for biomedical material sciences at the Institute of Biomedical Engineering. She is also a Royal Academy of Engineering chair in emerging technologies. Molly's research balances the investigation of fundamental science with the developments of technology to address some of the major healthcare challenges. The Stevens Group is a large and extremely multidisciplinary, highly dynamic research group using innovative bioengineering approaches to pursue their vision of solving key problems in regenerative medicine and biosensing. Molly's research has been recognized by dozens of prestigious awards, including the FEBS IMBO Woman of Science Award, and she is a fellow of 8 UK societies, including the Royal Society and the Royal Academy of Engineering. And she's an international member of the US National Academy of Engineering. She holds numerous positions leading research collaborations, publications, and organizations. So welcome, Molly. It's great to have you with us this evening.
Thanks so much, Alice. Great to be here.
Katya Pchelintseva is interested in multidisciplinary research, and her interest developed while she was completing her bachelor's degree in biology at Imperial, during which she participated in immunotherapy and tissue engineering projects alongside her studies. Eager to work in healthcare innovations, Katya changed fields during her MSc in biomedical engineering to work on a novel method for HIV detection. Recently, she further advanced her experience in diagnostics as part of the team developing an ultra-sensitive point-of-care test for COVID-19. Currently, Katya is a PhD candidate in the Stevens Group, where she is making new pattern models of heart tissue for drug screening applications. Katya is also an active member of the scientific community. She is co-chair of Future Investigators of Regenerative Medicine Symposium. She's an alum of our wonderful Imperial We Innovate program for women entrepreneurs. And she's an organizer of monthly professional development forums for the Stevens Group and a regular volunteer at Imperial's outreach efforts. So this will be a conversation, and we will see how these impressive women have come to be the scientists they are today and where they're heading. We welcome your questions, so please put your questions in the Q&A function, and our team will pull them together for our discussion. So let's start with Molly. Molly, you've made such many groundbreaking advances in nanoscience by pioneering the developments of nanomaterials and ultra-sensitive biosensors for disease diagnostics. Can you share with us one of your aha moments where you made a discovery from your science?
Thanks so much, Alice. I thought what I'd do is just show just a few slides about a little bit of a flavor of some of the things that we're doing. The aha moments actually to me are quite interesting because they change from when you're at the bench yourself. You know, I remember during my postdoc being the first person looking down the microscope, and you know, that's very aha when you get a big discovery. And nowadays, obviously, everything I do is team-based, and it's really the joy of seeing the team do well, and those kind of collective aha moments we have as a group. And that's some of the things I want to talk about today. So I'm just going to bring up a few slides. Hopefully you can see those slides. So yeah, as I mentioned, and the topic of this conversation today really is about thinking about how we can use multidisciplinary science, and in my case, how can we use it to make society better? And it is absolutely a team-based effort. You can see a picture of my wonderful group up there on the right. Really from so many different countries across the world, and also so many different backgrounds and many other types of diversity as well. And it's that diversity of skill sets and thinking, which I think enables us to have such a fantastic team. The other thing that we do, which is perhaps a little bit unusual in an academic setting, is that we have aspects of our research that focus both on fundamental science and also following that through to application, and so into innovations that can hopefully really help in patient care. And I find that fundamental side of it actually really inspirational for the work that we then lead into application. And one of the things that we're very interested in is thinking about how we can detect diseases earlier. And this can really have applications in a number of different spaces, but I'll just show some of them here. Some of the things we're interested in, for example, early heart failure. Can you pick that up earlier? Can you then treat patients earlier? Early cancer, of course, we know if you can pick cancer up much earlier, it's going to have a huge impact on patient survival. And very, very close to my heart is early infectious disease detection. And so we're working in all these types of areas. And we've set up a wonderful center called iSense, which is focused on a lot of these applications, and work very closely with collaborators, for example, in South Africa and also in Uganda. And this idea of thinking about how can we make diagnostics that are useful, of course, in the UK, but also across the world and in the developing world is something that really drives me. And in particular, I'm always thinking about how can we help in democratization of access to healthcare. And one of the things that's really exciting is thinking about how we might be able to develop diagnostics that work really, really well, but get them to the most people possible. And one of the ways we can do that is by making diagnostics, so essentially medical detection kits that can be used even in quite remote settings by being read by a mobile phone. And this is really, really important because there's now so many growing numbers of phones across the world, including in places like Africa. And if you think about that, what you can do is develop tests that might look, for example, a lot like a pregnancy test, except they work much more sensitively, so they can detect very, very early disease. And you can then take photographs with them with a mobile phone and use this to essentially detect the condition of the patient, but also potentially integrate this into online care pathways. And if I just give you an example of how we've applied this in HIV, we're interested in very early detection of HIV, so what we'd call the acute stage. And one of the tests that we've developed, the commercial test limit would be here, and our test can get right into that acute stage, so detect patients potentially a lot earlier. The second thing I want to mention is the skill sets we have, and this very multidisciplinary team we have, means that we can apply our engineering principles in many different kinds of application areas. So one of the other things we also look at is applications in regenerative medicine. And that might sound quite different to the biosensing, but actually it's all about those same skill sets coming together. So having very multidisciplinary teams with engineers and also with surgeons and medical people and chemists and lots of different people. And we're interested in applications such as making materials that can help to repair damaged hearts, can help to repair damaged knees, and really also help with regeneration in the eye, a very broad field of application. And we have a hub set up across the UK, which is part of the UK Regenerative Medicine platform. It's a hub for smart materials. I'm currently the director, and that is a hub that enables us to make biomaterials. So these are essentially materials you could put in the body and they'll enable cells to grow on them and then regenerate new tissue. And they can have all sorts of different forms. They might be like jelly, or they might be 3D printed structures. They might have different gradients of chemistry or very intricate architectures. And I'm just going to finish with just a very, what I think is quite a stunning picture of the sort of intricacies we can engineer into materials. What we're looking at here is a bed of very, very small needles. And on the surface of that is a single stem cell. And you can use the needles, which are man-made, to interact with that cell and to deliver information to the cell to really try and promote it to then go on and make a particular type of tissue regeneration.
These are such amazing studies and such beautiful images, Molly, and you're quite modest. It's really a huge advance to be able to detect something like HIV as early as you can. Perhaps you could say a little bit about how that extremely high sensitivity comes from your tests. And the other question is, what are the size of those needles on that picture?
Yeah. So in terms of the HIV, at the time we published this, which was a couple of years ago, this was the most sensitive point of care test in the world for early HIV. So you're quite right. You know, the team did a fantastic job bringing all that work together and that achievement. And the way we manage that is in a traditional pregnancy test, for example, you also have small particles called nanoparticles, which are normally made of gold, and that's what gives you the color at the test line, or indeed in the COVID test that people may now be familiar with. But our particles are slightly different in their chemistry, and they can essentially lead to an amplified signal. So the signal's even stronger, the test line looks even darker, and that enables you to detect diseases earlier. And then in terms of how small are the needles, well, we can engineer them to be different sizes, but if you think of a cell being maybe 50 microns or something, and you could get probably more than 50 of these needles per individual cell, so they're really quite small.
And you can make these exquisitely sensitive tests for cancer now, for some cancers, true. Is that true? And that early detection is just so important.
Yes, yeah, we've done a few different things for cancer. We are very actively working on what we call point of care tests. So tests, again, that would look a bit like a pregnancy test and looking at those in early cancer. We've also done some work which actually turns the urine blue once you add some chemicals, if you have an early type of cancer. And those needles actually that I showed you, we've also applied those to human tumor margins to look at enzymes that are upregulated in cancer cells with the aim of helping the surgeon to be able to demarcate much easier where the tumor margins are when they're doing a resection surgery.
That's fabulous. You're just at the nano scale, nanomaterials, just at the right size to connect to all these cells and all of these diseases as well as human cells. It's really fabulous. Katya, you must find it very inspiring to be in Molly's research group. You're in your final year of your PhD. And I'm interested in how you got interested in science and then perhaps the aha moment that led you into regenerative medicine. And we can talk a bit about regenerative medicine.
Yeah, of course. Thank you so much, Alice, and thank you for having me here today. Um, I think for me, my interest in science was kind of from very early on. My parents were both quite interested in science. They're both engineers, so we always had science around the house. But it's kind of the more mature time point when I became interested in science was when I was in Cyprus at the age of 16. And I was undertaking a placement in the hospital there, where I was able to watch all of these surgeries being performed on the patients for about a month. And I found that experience just so inspiring, because you could really see how the day-to-day work of those doctors was influencing lives of people. And I think it echoes in a little bit of a way what Molly said previously. I just find it so inspiring to be able to work in the field of healthcare. And that's kind of where it just started kind of blossoming, I guess. But from that placement, I knew I really wanted to stay close to healthcare, but I maybe wasn't very well fitted in terms of doing the surgeries myself, or it wasn't something that I wanted to pursue. So I was really keen to work at the forefront of healthcare and at the forefront of innovations to healthcare, and trying to see how we can drive what we already have in the current modern medicine to be better, and to kind of, achieve all of the novel therapeutics and novel regenerative medicines that we have today, and maybe push them further. In terms of the aha moment, I think for me, the aha moment has been certainly, it feels like it's been longer than just a moment. It feels like it's spanned across several years, and it's been a very long work in progress. But there are several defining points in my academic career so far, and I think one of them was in my undergraduate when I was in my second year, and I was working on writing a review paper with one of the professors here at Imperial, Professor Jamgas, and we were looking at how we can it was a literature review, and we were looking at what has been done so far in terms of modulating ion channels to drive stem cell differentiation. And I think that moment was really defining for me in several ways. And one of them was that I realized that I was capable of collecting all of this literature and reading, all of these concepts and putting them together and figuring out what is left there to be done and how something can be done better. And it really gave me an insight into a life of a researcher, but also just opened up this whole field of stem cells to me. And I realized that stem cells is not just something that we can study, but also something that we can manipulate. And it can be done so elegantly with biomolecules that are already existing, but also, like Molly mentioned, with man-made materials and biomaterials. And it can just be so fascinating how we can affect the behaviors of those cells and drive their fates and make them into into tissues that can be further used in regenerative medicine. So I found that just so fascinating, and I was really interested in pursuing that further on.
So we've talked a bit about the tests being very sensitive and that biosensing work, and you worked a bit on the COVID tests. How about with regenerative medicine? Can you tell us one of the examples of an interesting problem you think is getting solved by some of this new science and biomaterials and regenerative medicine?
Yeah, absolutely. I think in terms of regenerative medicine, the difficulty is so much in the fact that we need to not only kind of create the new tissue, but importantly to also replicate the niche within which those cells are residing. And that is very important for the regenerative medicine as a field, and is quite tricky because there is just so many components within the human body that are just so complex and are very difficult to replicate. So starting from something more on the chemical side of things, so whatever biomolecules are present in the tissue, but then moving on further to the structural components, such as alignment within the tissue. So for example, your muscles or your heart muscle has a very definitive geometry, and trying to replicate that on top of replicating the biomolecular components of the tissue, on top of replicating the cellular component of the tissue, is just, I think that is the biggest challenge and the biggest kind of interest in the field is how can we replicate it so faithfully that the resultant engineered tissue will not fail once you implant it into a patient. And there are so many ways in which you can modulate it using materials, and I think that that's just very fascinating and exciting field to be in right now.
That's really exciting. So you work from the molecular level all the way up through the cellular and then the macroscopic, because the shape matters if it's a knee or a heart or some piece of someone's body.
Yeah, absolutely. And I think it's also that this kind of scale is also one of the reasons why something similar to what Molly mentioned earlier as well, why sometimes even though people think biosensing and regenerative medicine are very different, but actually we can learn so much from the biosensing and from those really like small scale kind of nano biological interactions to then use that to inspire kind of larger macroscopic scale principles and use that for the regenerative medicine approaches instead. So I think that's why, particularly in our group, it's very useful to have both of those components. And in terms of regenerative medicine, of course, there are so many fine details and different levels and different scales within the tissue that are absolutely vital for regenerating a particular tissue.
That's really interesting, and let's bring Molly back in your research group, because Katya is just one of your many wonderful members of your research group, and you bring together people from different backgrounds because they bring different strengths and different fields of science together for these very complicated problems. And I think that some of the really exciting discoveries come from making analogies where you know something in one field and then you can apply it in another. And so I wanted to talk a little bit, maybe the both of you, but starting with Molly, about your vision for this multidisciplinary, multicultural team tackling these problems, and then, how that works and how you engender the right spirit so that you are so successful bringing these wonderful discoveries forward.
Yeah, it's worked so well, I think. I mean, I'm a complete convert's not the right word, but I'm completely sure that having these teams that are structured in the right way and that have just such wonderful people in them is the way that you can achieve the most possible. So in our group, it's really, really broad, actually. We have obviously a lot of engineers. We're in the Faculty of Engineering, but we have a lot of chemists. We have a lot of physicists. We have quite a few mathematicians now. We have cell biologists, and we have four or so surgeons. So it is about as broad as you might imagine. I really tend to take people based on how brilliant I think they are and how well they'll fit within the team structure. But I look at it really from a high level of how the different skill sets can come together for the different sorts of projects that I'm thinking about. And if you don't mind, Alice, I'd quite like to just show one picture of
show us your team. You have some beautiful pictures from your OA days where you're out in some mountains hiking or something.
Oh, we do. And we also have this. I don't know if it's going to let me play. Oh, it's not letting me bring it up. But we also have a really interesting periodic table of interdisciplinary members of our group that was put, that I'll just share with you for, oh, that's sharing the wrong thing, isn't it? There we go. That was put together for a celebration we had a couple of years ago. And this is people from the current group, but also our alumni. And what we did was look, in very sort of broad terms at some of their background. And you can see there those sort of key themes coming through around the engineering, the biology, medicine, chemistry, and physics. And arranged. I don't know why I'm purple, actually. I think that just means I'm a mix of different things. But you can see really the sort of huge diversity that we have, and it's really all those brilliant people coming together that means we've been able to achieve the things we've been able to achieve. And you know, Katya's a wonderful example of the sort of high quality of people that we get within the group, but also that have a very, very collaborative nature. And that's one of the things that I'm very careful about with my hiring when I bring people onto the team, is having people that will really support each other and have a nice group structure, at least what I hope is a nice group structure.
That's marvelous. I'll have to inspect it to see who the noble gases are later. It's a great way to show the diversity of your team. And you had an earlier image with everyone having flags of all these different countries. And I have a theory that people coming from different cultures also bring different aspects to their science because of the way they grew up and what they learned. And maybe you or Katya want to talk a bit about the international aspects of your team.
Yeah, I'll let Katya have a go, and then I can maybe add some comments.
Yeah, I think it's actually quite an interesting point that you know, it's not only multidisciplinary but also very multicultural in the Stevens group, and I think that just makes every day you know, of your PhD here so completely different and so interesting, because you interact with those people as colleagues but also as friends, and you just learn so much from them and from their life experiences. And I think it is those kind of life experiences shaped by our cultures that also lead to us thinking about the same problem maybe differently, and it just helps to kind of have this completely collaborative environment where we're never short of ideas. You know, no problem is too big. Sometimes when you think that you're stuck on something, you just need to talk to your colleagues who will be able to you know, see it from a completely different angle. And it is such a spectrum of different scientific approaches to the same problem. And I think I can kind of draw from my own personal experience from, you know, I was born in Russia and I lived in Russia for the first 16 years of my life. And throughout education there, we kind of almost always relied on a very analytical approach, and it was brilliant, and the tools that we had were fantastic, and the people were just completely, you know, amazing at doing what they were doing. But there was maybe not as much space for creativity as I've noticed that we have here in the UK once I've moved here. And I think sometimes you do need this kind of combinatorial approach where you kind of draw from the different experiences, both discipline-wise, but also culture-wise, where maybe your colleague knows how to approach the problem completely differently to how you would do it yourself. And I think in Molly's group, it's completely like that. And it just makes collaboration so interesting. And it kind of teaches you how every problem that you are trying to approach, or any new project that you're starting, there will always be a novel angle to it, from, you know, looking at it from maybe an engineering perspective, or more a physicist perspective, or a chemist perspective. And it just makes research so much more interesting, and in a way also more fun.
That's great. And your group is so impressive. When we were talking earlier, I learned about some things you're doing that really help your group members well beyond just the scientific collaborations, the things that keep you a well-rounded and bonded group. I wonder if I wanted to copy a few of them, I think college-wide, and maybe you could share a few of the things that you do within the group, because it's such an important part of your life, working in a group like that. And it sounds like you do some really wonderful things. And meanwhile, the questions are pouring in, so we'll get to the audience questions. But I just wanted to finish off with a little bit about your group and beyond just the science.
Yeah. We'll come on to some of the pastoral things in a second. I'll hand over to Katya actually, because she's been helping out with some of those. But I would say it's a very social group. I mean, you mentioned earlier our sort of group retreats, you know, our science retreats, where we go and sort of brainstorm science, but also go hiking together. The image that had all the flags actually was a dinner that we did, which was sort of foods of the world themed, where everybody brings food from the different countries. So if you're someone like me that, you know, just loves eating loads of different foods, it's kind of a paradise, really. And we try and do those quite often. But we've also recognized that in science in general, you know, it's really, really important to also have training in the things that are not just when you're at the bench, you know, the sort of more well-rounded side of things. And we've set up this wonderful forum called Fab Forum that we run for our group, which Katya's now leading. So I'll hand over to her and she can tell you a bit about it.
Yeah, and absolutely, Molly, I agree with the fact that, you know, being a scientist is so much more beyond what you do on the day-to-day in terms of experiments and, you know, experimental design. It's so much of being also a good communicator, or being able to do quite beautiful graphic design for the figures for the paper, or even, you know, how would you write the paper? So you need to be a good writer as well. And it's all of those skills that you kind of almost try to acquire along the way, but very often there is no formal way of finding those resources for individual researchers. So in our group, we use Fab Forum for things like that, which is basically a professional development webinar at the moment. We used to do more in-person meetings, but it's really just quite an informal space for all of us coming together and for us to discuss you know, the topic of the week. So it's kind of determined by what the needs of the group are at the time. So quite often we'll ask for feedback from other members of the students group to say what is the skill sets that they feel like they would like to acquire. And we've had just such an amazing success with loads of people in attendance. And I think people find those discussions very useful, because that is really the space where you can say, actually, I don't really know about this topic so much. Can, like, you, my colleagues, try and help me learn more about it? And it's really a great place for us to connect also on a social level. Sometimes we run more of a kind of pastoral care topics, such as, for example, imposter syndrome. We talk about that quite a lot. And it really, like I said, it is very informal and more for all the researchers to come together and discuss what does it mean to be a researcher and what skills would you like to acquire. And we also have done a few kind of career-related forums too, where we discuss about different kind of things you can do once you have done a PhD and different opportunities that there might be. So it's a very, I think it's very effective in making all of us come together as not only colleagues, but also a community, and be able to support each other during those sessions. So it's really a valuable place, I think, for everyone.
Okay, thank you. And actually on that career piece, we've got such a strong alumni network from the group, and we often have alumni reunion events, and the alumni will always come back and talk to the current people in the group about their jobs and you know, their route to getting there, and the sort of different types of career you can have with a science background. And I think that's been something that the current people in the group find invaluable, and of course is really enjoyable to the alumni as well.
I was just going to bring up the alumni. It speaks to one of the questions that we've had, and then we can go to some of the science questions. But the comment that it's a challenge to maintain the longevity of multidisciplinary teams, given that everyone has their specialization and they want to have their own career track. And, you know, I thought that your convening group and keeping in touch with the alumni it does speak to that a bit. And also how people can balance that, that specialization and their expertise with collaboration with people with other specialties and such. And I don't know how much ebb and flow. Obviously, your students come through and graduate, and you have other collaborators come and go, but it seems to be a very healthy, dynamic organism. Yeah,
it's wonderful. I mean, it's always changing, because people probably stay on average 3 or 4 years in the group. So on any given year, you might, you know, have a 25% turnover or 30% turnover of the group. So it's changing, but it doesn't change all in one go. It's a sort of gradual people leaving and going on to fantastic jobs, and then other new people joining. And we've got our alumni, you know, 45 of them are now professors, and many others have just got fantastic jobs in industry or have set up companies, including, you know, exceptionally successful companies. And many of the alumni in their sort of next life after leaving the group are collaborating with each other, which is really exciting to see as well. And oftentimes they reach back out, and we end up doing joint things down the line as well.
So there are a few science questions that have come in. Have you ever looked at exhaled breath as a method of collecting samples? From Tanya Jameson.
We haven't, but we're very interested in that. We have a technology which is a little bit different to the ones I talked about, but which is essentially designing a little device that could measure potentially blood from the patient, but also potentially down the line could also measure breath. And I think that is really interesting in terms of looking for early disease markers.
Another question says, great program from Mustafa Dangos. Do you think the nanoneedles can be used to detect electrical signals in tumors, remembering that some electrical signals occur very early in the cancer process?
That's interesting. The work we've done so far with them, with tumors, has not been with electrical type needles, but we do have a portion of our group that is interested in this field called bioelectronics. And one of the things we can do there is to actually make these very small needles able to detect essentially conductivity. And actually maybe I'll pass to Katya, because she's doing some interesting things with bioelectronics as well in a slightly different area.
Yeah, I'm afraid I haven't done electronics in terms of kind of disease detection, but I think that definitely it would be really interesting to look at the nanoneedles from the kind of conductive perspective in terms of detecting the electrical signals within the tumors. I think that would be something that would be a really cool and quite an elegant way of detection. But I'm afraid I haven't done that myself.
I was thinking more of your sort of the interface of materials with heart tissue and the potential role that you could have there in the future with bioelectronics.
Yeah, absolutely. There's so much in terms of doing bioelectronics and cardiac tissue engineering, and of course the heart is very electroactive, and there is quite a few people in the group now who are working on design of novel kind of materials which are conductive, and we're kind of currently working on using those as a platform for delivery of stem cell derived cardiomyocytes to the heart. So that is something that is quite close to my heart.
Great. And this actually, there's a question probably for Molly, but how long does it take your research to go from lab to market?
Really depends entirely on the application. We are at different stages for many of the different innovations. So some of them are already being tested with human samples. Others are much more in the sort of innovation earlier stage. In general, if you have an invention that needs to go inside the human body, that's going to take longer. And if you have obviously something like a therapeutic drug, that might take even longer still. But if you have something that is just testing body fluid, but is not actually going inside of the human, then that route to market is definitely quicker.
So we have, I think, quite a few school students and others on call, and it would be nice to hear you all thinking, both of you thinking back to when you were a youngster, and what kinds of, what advice you might give them, how they can get active and involved in science as they're going through school and moving on to university. And then perhaps we move then to university and PhD. But what is your advice for the young people on our call today?
Katya, do you want to go first?
Yeah.
Since you're younger, it's more recent that you were at school than I was.
I think if I was to give advice, it would be something along the lines of, you know, if you're just curious about something, that is reason enough to pursue it. I think quite often it's seen that, you know, to be in science, there needs to be quite a big precursor or quite, you know, a very strong interest in something in particular. But sometimes just, you know, just being curious about something small can be the first step. And then there will be something else that you'll be curious about, and that will kind of guide you through your journey into science. And I always found that for myself, it was always more relatable when I could think more kind of short term, kind of what is it that I'm interested in right now. And I'm always kind of keen in exploring different areas that I haven't really had experience in before. So I think that that's something that I would kind of advise like someone over school age right now, that it doesn't need to be this, you know, huge thing that you are like super interested in doing. Just be curious and follow your interest.
I think that's really good advice. I think one of the things that's the most amazing about working in science is firstly the ability that what you do could eventually help people. I think that's really, really motivating. But also the fact that you're just never ever bored. You know, there's so many things to learn. I mean, the only thing I know is how much I don't know. You know, there's so much still, so much to learn. It's really impossible, I would say, to get bored if you're actively researching in science, because there's always going to be things you don't know. The field is always moving forward, and so in that way, it's just always so interesting. And I think, to me, that lack of boredom, you know, as someone that just enjoys learning so much, it just provides that opportunity to be constantly stimulated.
And you both evolved in what you are most excited about. And I think that's being open to, as Katya says, your curiosity. It'd be interesting, Molly, where did you start your studies, and did you picture yourself being where you are now? Or were you thinking about something else?
I didn't picture myself being here. I'm really, really glad I'm here, but I feel that you know, just constantly blessed to have such a great research group and such an interesting job. But I actually started off doing pharmacy as an undergraduate. I actually was perhaps more motivated by medicine, but I didn't want to spend 5 years at university. I thought that was far too long, and which is so ironic because I've been at university ever since. But you know, that's why I went for pharmacy instead of medicine, and it was a really, really interesting degree. But I knew that I wasn't ever planning to work as in a community or hospital pharmacy. I was very, very interested in research, and so I then did a PhD in single molecule biophysics, purely because I wanted to really push myself and do something I didn't know anything about, and just learn as much as possible. So it was really just, to me, an intellectual challenge, the reason I chose my PhD. And then my postdoc was in a much more applied field. So it was in regenerative medicine, and that was firstly very lucky that my supervisor took me on, because I was coming from a very different background, but it was very much motivated by wanting to do something where I could see the impact that would have on people and how it could help them.
That's great. And it sounds very similar that Katya's on a similar path, interested in medicine and where to make a difference, but didn't want to be a medic.
Yeah, it does feel like, you know, I'm very honored to have some similarities in my path as they are in Molly's.
So it's I think it's important for people to recognize that there are opportunities to keep your options open and your eyes open so that you can learn from all these multidisciplinary colleagues, and it shapes your own path and what you're able to do as well.
Yeah, I also think, you know, maybe this is advice for the young folk as well, that, you know, just keeping open-minded about things. And, you know, sometimes people have such a linear view of where they need to go. And I think that can sometimes shut quite a lot of doors of opportunities if you do that. I mean, it's of course good to have a long-term vision for the impact you want to have, but the route that might take, you know, will depend a little bit on the opportunities that come your way. So I think to me, keeping a really open mind and being really agile when different opportunities came up, and, you know, working really hard. But if you work hard at things you enjoy, it's more like a hobby than what you'd classically define as work. So definitely, you know, really putting your heart and soul into it, but in a format that you're going to find enjoyable is just a great way to have a career that's you're going to find fulfilling.
Thank you. That's wonderful and very inspiring. There's another technical question that may spark some people to think about some other work. Have you looked at all at bioinformatics and specific genes and their possible links to cancer susceptibility or cancer suppression?
We are really interested more and more actually into how we can bring aspects like bioinformatics, but also many collaborators from computer science and from the statistics centers at Imperial to be part of our team to help us in the gene profiling, as is expressed in that question, but also more broadly, you know, how do we use elements of underpinning big data essentially to help guide the sorts of experiments that we want to do. So it's something we're thinking about all the time and really, really interested in.
Great. And another one, are you interested in using nanoneedles for antibacterial and antivirus applications? From Pavani Chirukapalli. It's
a very interesting question. We are, and we've done a bit of work in that area, which isn't published yet, but it will be published at some point. In general, there's definitely potential there, but we have to bear in mind that the size scales are different. So things like viruses and bacteria are much, much smaller than human cells. And so the challenges around the engineering are a little bit different once we start looking at those other applications.
There's a related question, perhaps. Have you investigated technologies for delivering vaccine antigens for prolonged periods of time without repeated needles? Faith Azir asks.
We are very interested in that. We're part of the Imperial Vaccine Hub, or rather the Vaccine Hub, of which Imperial is one of the leaders with Professor Robin Shattock. We also are working really closely with the Gates Foundation. So one of the things I'm interested in is working in many different ways to try and achieve impact. And one of those ways is working through not-for-profit organizations such as the Gates Foundation. And with the Gates Foundation, we have some long-term projects that are around the development of what we'd call next generation type of vaccines, and also long-term implants that can help with other disease conditions as well.
You mentioned the Gates Foundation, and I think of the incredible job you also have, Molly, of raising all the funds to support this large multidisciplinary, multicultural, very active group and all the research. And you have very strong funding from the UK government, from European sources, but also from foundations and philanthropists. And I've been thinking a lot about how important this unfettered blue skies funding is, so that young people like Katya can pursue new ideas when they're starting their career. We have junior fellowships, Imperial fellows. We're trying to support early career academics so they can have some time to just do the science and not be fundraising. But it'd be interesting to hear your thoughts on how, how you raise funds and what you're thinking about in that balance of showing impact and applications with also pursuing just totally new off-the-wall ideas.
It's interesting. I think the funding structure in general is that it's quite important to be able to show translation and impact for some sources of funding, and then there's other sources of funding available that are a bit more blue skies. Some of our very high impact blue skies fundamental research actually quite often has started as side projects, so that were maybe something that was done slightly on the side and actually turned into a really exciting discovery. And a lot of the funding that we have at the moment has quite clear impact in mind, and that's really because I'm really interested in making sure that some of the things we're developing that can be useful do end up being useful. But we also have really exciting fundamental research like the discovery of bone structure that we published recently with York University in Science, and many other discoveries like that are definitely purely fundamental, but they're just fascinating science.
Yes, and those discoveries do ultimately lead to these important applications as one understands the progress of a disease or how a material or a
they're
tissue
really important.
Yes.
Really important. And I think it's really key that the funders create an ecosystem where that sort of research is also really well supported.
So there's a question for Katya here from Maeve Wong. Have you thought about your next destination after your PhD? What are your plans, Katya?
Hi, Mai. It's great. It's a great question. I have been thinking about it quite a lot recently, especially because I'm in my final year now. And I think that's also something that is really great about having a PhD in such a multidisciplinary group like ours, is that there's just so many possibilities that you can do and so many opportunities that you can pursue after your PhD, that I kind of really wish that I could split myself into several people and do all of these things. I think I would be quite keen to try myself out in something more closely related to industry and to really see, how all of these, regenerative medicine concepts and regenerative medicine kind of constructs that I'm developing could be actually used in healthcare, and kind of work on this interface from bench to bedside. I think that would be really great for me to get experience in, but I'm not limiting myself with anything right now, because I feel like especially at the end of my PhD is when there is just so many opportunities, and like Molly said, I keep being open-minded and just seeing what is the right next step for me.
I think the thing is, when you have such great students like Katya, that they're so talented, and they just could do so many different things. And we've had students, our students have just done fantastically well on leaving the group, and some of those have stayed as doing postdocs in either closely related areas or something completely different, and others have set up companies immediately on leaving the group, and others have joined industry or not-for-profits. And really, you have so many options that it comes down in the end to what you think you'll enjoy the most, and often it comes down to looking at what those opportunities are, and just going a little bit with your heart as well as your head, I think.
That's great. And that both of you, it brings up to mind, translation isn't just directly from the bench to the bedside. It often needs a corporate partner or a spin-out company to help develop the technology and be able to get it into medicine. And so that translation from university to hospital often involves partnerships with small companies and large companies, and that's a really important collaboration.
Yeah, absolutely. And we work, both with industrial partners, but also in the setting up of NUCOs as well.
So we're getting to the end here. There's one more question on how you achieve work-life balance in the midst of your busy schedule. I know you have children, Molly, and you take great care and great pride in them. You want to share some ideas?
I don't know. I don't know. I mean, I think, as I mentioned earlier, making sure that you're enjoying what you do means that it's invigorating rather than exhausting. So that's always a good hint. I mean, I find actually, my interactions I have with my group are just energizing, actually. I mean, they're just such a wonderful source of inspiration to me, and energy. I'm super physically active. I run all the time and I do yoga all the time, and so I'm very kind of conscious about making sure that I keep my body really healthy, because I think that's also just gives you more energy, and then making sure that I do have time for my kids and my friends. But I have a busy life, that's no question about it. And Katya too, right?
Katya, yes, Katya, what do you do?
Katya works hard and she's social. Yeah.
Yeah, I think that's exactly kind of similar to me as well, Molly. I find interactions within the group so energizing, and it really does help so much that we're not only colleagues, but we're also friends, and we see each other socially outside of the lab. And it's just so helpful in terms of keeping that work-life balance, because it's not a chore to come to work. It's really such a pleasure to be here, and it's just so nice to interact with like-minded people and have all these socials that we have. And I also try to really keep busy kind of outside of the lab, but also kind of still involved in scientific kind of field, but in a kind of different way. So I try to like we mentioned in the beginning, run some conferences from time to time, or participate in the We Innovate program. And I think that really helps me see my research from different kind of perspectives, from the entrepreneurial side, from more kind of event organizing side, but also to kind of have fun with it. And I think that's really the way forward. So it's not seen as something that you do because you have to, it's something because you do that because you enjoy it. So
thank you, Katya and Molly. Your passion for what you do really shows through, and it's so exciting to be able to talk to you. Thank you very much for this evening. And I want to thank the people watching and our friends and benefactors who support PhD students like Katya and research programs like Molly's. Our next conversation is on Tuesday, May 25th. It's called and entitled, "Is There Such a Thing as a Free Lunch? How Economics Helps Us Understand the World?" With Professor Dame Carol Proper and Dr. Mirabelle Mules, two inspirational Imperial women in economics. So we look forward to seeing you then. The We Innovate, I think, awards, or the We Innovate pitches are on June 25th, I believe, and I'll also make sure this audience finds out about that. So thank you all, and good evening.
Thank you.
Thank you so much.